Hello students, this is Molecular Basis of Inheritance Part 2.
So in the earlier session, we have studied what are the purines, pyrimidines are these bases, okay, and the chemical components of nucleotide. What is the difference between a nucleotide and nucleoside? We'll try to understand on the basis of that what is the structure of how the structure of DNA is made up or produced or formed by these kinds of bases that we are going to study in this part, okay. So in the structure of DNA, it is very essential that we have to understand thoroughly that what is a DNA molecule, and a DNA molecule is a double helix. It is a double helical structure of each strand. You must understand what are the complementary base pairings, polarity of the strands. Now we are going to discuss it, and the major minor groups. So we are going to discuss it one by one, but before going to get into that part, it's you can observe that there are the two diagrams, okay. So this is the first diagram where it is the diagrammatic representation of DNA molecule, okay. Now you can observe that these are the two arrows, okay. There are two arrows. Now this arrow is ascending strand and another is in opposite direction, and this is descending strand, okay. So this is ascending strand, another is descending strand. Now here we are talking about the strands of the DNA. What these arrows? These are the strands of the DNA, okay. Now try to understand this part. We have studied this part in the earlier slide, if you remember, okay. So this is the sugar, this is phosphate group, and this is a nitrogen base, right? Now observe the first part. Phosphate group, it is denoted by 5 prime or 5 dash end, okay, while this is sugar, and sugar is denoted by 3 prime or 3 dash end, okay. So if you observe this part, these are the sugars which are having these phosphate groups. Now see, these phosphate groups, they are attached, okay, they are linked. Now the sugar phosphate is attached like this, and it forms a part of ascending strand of DNA. Another part is these are the nitrogen bases, okay. In the ascending strand, observe these nitrogen bases. This is adenine, another is guanine. Now these are the rings, double ring and single ring. If you remember the structure of purine and pyrimidine bases, so purines and pyrimids, single ring and double ring compounds, so all they are denoted by this. Adenine is double ring compound, okay. So that's why it is denoted by a double ring compound, okay. Thiamine, adenine gets attached. Now see the bonding. These are the double bonds. A pairs with T of other strand. Only adenine, guanine, thymine, cytosine and adenine, up to this part, this is one strand of the DNA. Are you getting that? This is the one strand of DNA, okay, and that strand is ascending strand. Now this ascending strand is paired with the another strand of the DNA, okay, and that another strand is descending strand. Now ascending strand and descending strands, they are paired, okay, they are paired by this chemical bonding. There is a pairing of adenine with thiamine, guanine with cytosine. See, double bond, triple bond. Can you observe that in the diagram? Thiamine is paired with adenine, cytosine is paired with guanine, and adenine again is paired with thymine. Okay, so this is the bonding. Now thymine, cytosine, adenine, guanine and thymine, these are the nitrogen bases of another strand of the DNA, and that strand is the descending strand. Okay, Now, this is the nitrogen base, sugar, and the phosphate group. Again, the phosphate group is attached with the sugar. Phosphate group is attached with the sugar. Okay, so this is another part of the DNA. This is another part of the DNA, and this is another half part of the DNA. Okay, ascending strand and descending strand, and these two strands, they are paired with each other by the pairing of nitrogen bases. Okay, so that's all about this diagram. Let's move towards the another diagram now. Now this diagram is colorful. You can observe these are the two ribbon like structures, if you remember, if you observe. Okay, this sky blue colored part. Okay, this is another strand, and the next ribbon is another strand of the DNA. Now they are forming a different kind of structure. If you observe the groove means a notch or the space, you can observe that. So it's a major groove and it's a minor groove. So see, observe the space. What is the difference between major and minor groove? If you observe the space between the strands, okay, the twisted structure when it is getting formed, it's a twisted ladder like structure. Okay, if you just imagine the ladder, and if you twist that ladder, so that type of structure, when it is twisted ladder like structure, whatever it is, so that type of structure is nothing but the structure of the DNA. Okay, so it's a major groove. Major means the space is more, minor means the space is a little bit. Okay, so it's a major groove, major notch, and minor groove, minor notch. What are the nitrogen bases? Now see, whatever we have studied in this black and white diagram, or the chemical components like diagrams in the form of these alphabets, this is shown diagrammatically, colorfully, how the ascending and descending strands, both these strands of the DNA, they are attached or they are paired with each other by this or through these nitrogen bases. Try to understand that now. Adenine is denoted by red color, thiamine by orange, guanine by blue, and cytosine by green color. Okay, now these are the base pairs of the DNA. Okay, and this, whatever this part is, blue colored ribbon like part, that part is nothing but the sugar phosphate backbone. Okay, now what is a sugar phosphate backbone? If you observe this diagram, now see, this is sugar phosphate, sugar phosphate, sugar phosphate. Now sugar and phosphate, they are forming the sugar phosphate backbone. Same with the ascending link, sugar phosphate, sugar phosphate. So this is the sugar phosphate backbone, means this part. Okay, and this ATGC pairing, that pairing is denoted by this ladder like structure. If you observe the colors, A pairs with T, red orange, AT, GC, blue with green, if you observe it carefully. Okay, so it's a twisted ladder like structure. So let's try to understand now. If you have observed this diagram carefully, if you have understood what the how the nucleotides or nucleosides, they play a very vital role in formation of structure of DNA. If you understand diagrammatically, let's start, or let's begin with the brief introduction or brief information of all these points now. So, starting with this first DNA molecule as a double helix, in 1953, James Watson and Francis Crick, they proposed the DNA structure based on the X-ray crystallography technique, which was provided by Rosalind Franklin. We have studied it in the introductory part. So, DNA molecule, they proposed that DNA molecule as a double helix. So according to this model, which was proposed by the Watson and Crick, a DNA molecule consists of two long strands. They are coiled around a common imaginary central axis to form a double helix. It looks like a twisted ladder. So you observe this diagram now. what they proposed, Watson and Crick, what they have proposed, that the DNA molecule, it consists of two long strands coiled around. Now these are the two strands which are coiled around a common imaginary axis, okay, and which it forms a twisted ladder like structure. Now the structure of each strand of the DNA consists of number of nucleotides. We have studied it. Each nucleotide is made up of deoxyribonucleic sugar, phosphate group, and a nitrogen base. The successive nucleotides of the same strand are linked by 3', 5' phosphodiester linkages, that is phosphate attached to the fifth carbon of a sugar of the nucleotide is joined to the third carbon of another. So we have studied it that phosphate and sugar, they form a backbone. So structure of each strand, it is made up of, or it is composed of nucleotides. It not only contains the sugar and phosphate backbone, but it also is composed of this nucleotide bases. Okay, so the successive nucleotides of the same strand, they are linked. So because of this type of attachment, now see, this is the 5' end. They are having two types of ends. It ends here, so this end is free 5 phosphate group. So it is denoted by 5' end. That's why it is 5' or 5'. Another, it ends with this attachment, that is 3' or 3' end. Okay, so it's the fifth carbon and it's a third carbon. So depending on the position of that, it is denoted as 5' and 3'. Now what is complementary base pairing? As I have explained it earlier, so the nitrogen base is present in the one strand paired with the nitrogen bases of the opposite strand. Now see, A, G, T, C, A, they are paired with T, C, A, G, A of the other strand. So there is complementary base pairing, okay, between the two strands. So purine base A always pairs with the pyrimidine base T, that is AT, adenine and thymine by 2 hydrogen bonds, and the purine base G always pairs with the pyrimidine base, that is C, cytosine, okay, with 3 hydrogen bonds. So AT with the double hydrogen bond, and guanine cytosine with the triple hydrogen, 3 hydrogen bonds. So this pairing is termed as complementary base pairing. Such type of pairing which you have observed in this diagram or in this diagram, that type of base pairing of this nitrogen bases is known as complementary base pairing. So that's all about this complementary base pairing of the DNA. Next part is polarity of the strand. So what do you mean by polarity of the strands of the DNA? So each strand of the DNA has a specific polarity. Each strand has its specific polarity: 5', 3', 5 to 3', 3' to 5', or 5' to 3', 3' to 5'. So their polarity is different. So they have specific type of polarity. One end is considered as 5' end, whereas another is considered as 3' end. So this polarity is because of third and fifth carbon atoms of the deoxyribonucleic sugar. If you observe, I have already mentioned it. This is the 5 carbon, fifth carbon sugar of the one strand. This is the third carbon sugar. Okay, sugar, third carbon position of that carbon atom of the sugar. Okay, so it's because of that. So third and fifth carbon, here we are talking about the carbon position of the sugar which is present in that strand. So at the 5' end, there is a free phosphate group. You can observe at the 5' end, there is a free phosphate group, while at the 3' end or 3' end, there is a free hydroxyl group or free OH group. So one of the strands runs in the 5' to 3' direction, and another strand runs in opposite direction, that is 3' to 5'. So it is 5 to 3, 3 to 5. So they are arranged in that opposite way, and that type of arrangement of the two strands of the DNA is considered as anti-parallel, due to which 5' end of one strand, or it lies close to the 3' end of the other strand, and vice versa. So this 5' is near to this 3', and opposite end, this is 5' is near to this 3'. So that is the anti-parallel polarity of the strands. So that's all about the polarity of the strand. So this is very important. Here you just talk about the positioning of the carbon atom, which is the free end of that strand of the DNA. Okay, so that's very easy to understand when you understand the structure of the DNA. Okay. Now, last point is major and minor grooves. So, what are these major and minor grooves now? Normally, the strands of the DNA, they undergo right-handed coiling. Now, this is my right hand. So, normally the DNA, they are right-handed. They undergo right-handed coiling around a central imaginary axis. Now, this is the central imaginary axis. Now, around this axis, the right-handed coiling, it occurs. Okay. So the coiling of the double helix results in the formation of major and deep grooves. Now these are the major grooves. This is the deep groove, and the twisting of the two strands around the another forms the minor or the shallow grooves. This is the deep groove, that is known as or mentioned as major groove. Another is minor groove, which is mentioned as shallow groove. They are found in alternate manner. Okay, so let's try to understand what is right-handed and what is left-handed coiling of the DNA. So how these grooves they are getting formed. So this is my right hand. Try to understand it. And this thumb is the central axis. Okay, this is the central imaginary axis, and this is the right hand. Now this is the coiling. This is a right-handed coiling, and this is my left hand. Again, this is the imaginary central axis. This is my left hand, and now it is another kind of coiling of the DNA, that is left-handed coiling. So this is right-handed coiling and this is left-handed coiling. So what exactly it is? So if you hold it pointing away from you and it twists clockwise moving away, it is right-handed. Otherwise it is left-handed. So the helix of normal DNA is right-handed. Now normally the helix of the DNA is always right-handed. Left-handed helices, they have been produced experimentally, artificially, and may be present in the living cells. Okay, so right-handed helices or right-handed coiling you talk about here, mainly it is present. Okay, so it is the clockwise twisting of the DNA. Another very important part of the components while studying the structure of DNA is what is the purine and pyrimidine ratio. What is purine and purine is to pyrimidine ratio, that is A plus G is equal to T plus C, or A plus G upon T plus C is always equal to 1. So what is that? So due to complementary base pairing, total number of purine bases is always equal to the total number of pyrimidine bases. See, purine and pyrimidine bases. So their amount or their number is always equal. So that because of that, the ratio is 1:1. So this is called as Chargaff's rule. 1:1. A plus G is equal to T plus C. The amount of purine and pyrimidines is always equal. Their ratio is always 1:1. So that type of statement, that type of rule is known as Chargaff's rule, and it may be represented by this equation. Okay, so that's all about purine is to pyrimidine ratio. So in this part, we are going to talk about the DNA as the genetic material. So it was not proved earlier. It was considered earlier that proteins are the genetic material. So there were few experiments which were performed to prove that DNA as the genetic material. So let's try to understand it. By the early 1900s, geneticists, they knew that the genes, they control the inheritance of the traits. Okay, so genes, they are present on the chromosomes and they control or the traits or the characters. The inheritance of it is also controlled by the genes. So that the genes, they are located on the chromosome, and they are chemically chromosomes, they are mainly composed of DNA and the proteins. Okay, so initially most of the geneticists, what they thought that the proteins are large, they are complex molecules, and they store more information needed to govern cell metabolism. So it was assumed that the protein caused the variations observed in the within the species. Okay, so in comparison to DNA, proteins were considered as the genetic material by few geneticists, okay, or by few scientists. On the other hand, DNA thought to be very small, simple molecule whose composition varied little among species. Okay, DNA, its composition, it varies. So over the time, these ideas about DNA were shown to be wrong. In fact, DNA molecules are large, and they vary tremendously within and among the species. Okay, so variations in the DNA molecules are different than the variations in shape, electrical charge, and function shown by the proteins. So there are two different kinds of variations uh, which were studied in the proteins, and or which are found to be present in the DNAs and in the proteins. So it is not surprising that most researchers, they initially favored protein as the genetic material than the DNA. So over a period of roughly 25 years, geneticists, they became convinced that DNA and not the protein is the genetic material. So there are few experiments which were performed by a few geneticists or few scientists. So you are having these three experiments in your syllabus that we are going to study, or that we are going to discuss in the next session. So that's all about the genetic material as the DNA. So that's all about today's session. Thank you.